Welcome to this lecture on early immune system theories. Today, we are going to explore a fascinating historical puzzle. How does our body know exactly how to fight off a disease it has never seen before? We will journey from early, incorrect assumptions to the elegant biological reality that keeps us alive today. By the end of this session, you will understand the foundational principles of modern immunology. Now that we have introduced our topic, let us start with a simple analogy. Imagine a locksmith trying to make a key for a mysterious, never-before-seen lock. Does the locksmith look at the lock and mold a blank piece of metal to fit it perfectly on the spot? Or does the locksmith already have a billion different pre-made keys in a massive warehouse and simply tries them one by one until one finally clicks? This is exactly the puzzle early scientists faced when trying to understand how our bodies fight off new, unfamiliar diseases like bacteria or viruses. This puzzle led to our first major historical model. The instructional theory, championed by prominent scientists like Linus Pauling, proposed a very intuitive idea. They suggested that an invading antigen acts as a physical template. According to this model, the immune system takes a flexible, generic antibody and physically molds it around the antigen to create a specific fit, much like molding clay around a coin. While this made logical sense at the time, it assumed that the body custom builds its defenses entirely on demand, which we now know is not how the biology actually works. Moving away from the idea of molding antibodies, we arrive at a competing idea. In stark contrast to the instructional theory, the selective theory was originally proposed by Paul Ehrlich as the side chain theory. He suggested that immune cells already possess a wide variety of pre-formed receptors on their surface, long before they ever see the antigen. According to this selective model, the antigen simply binds to the specific receptor that happens to fit it by random chance. This binding event acts as a trigger, thereby selecting that specific cell to produce more of those exact receptors. Building upon Ehrlich's foundation, we reach the accepted biological reality. Ultimately, the selective theory triumphed and was beautifully refined by Frank McFarlane Burnett into the clonal selection theory. This theory states that each lymphocyte, or white blood cell, has a single type of unique receptor on its surface. Upon binding an antigen that perfectly matches its receptor, that specific lymphocyte is stimulated to divide rapidly. It does not just make more receptors, it makes more of itself, creating a massive clone army of identical cells dedicated entirely to destroying that specific microscopic threat. To truly grasp this mechanism, let us look at a visual representation. On the left side of this diagram, you see a diverse population of naive B cells, each equipped with a uniquely shaped receptor. Notice how the triangular antigen only binds to the one specific cell in the middle that has the matching receptor. This is the selection phase. Once selected, the arrow shows this single cell undergoing clonal expansion, multiplying into a massive cluster of identical cells. All of these cloned cells now produce the exact same antibodies, perfectly tailored to neutralize the invading antigen. Now that we understand the biological mechanism, we must ask why this matters today. Understanding these early theories is absolutely crucial because they form the bedrock of all modern immunology and medical interventions. Because the clonal selection theory is the biological reality, we know that the body keeps a memory of pre-selected cells ready to fight a second infection. Furthermore, This concept is the direct foundation for producing monoclonal antibodies in the laboratory. These are life-saving treatments used today for cancers, autoimmune diseases, and severe infections. Modern immunotherapy would simply not exist without this knowledge. Having seen the biological importance, let us look at how we can quantify this process. While immune theories are highly biological, the rapid multiplication of selected cells can be perfectly described using the mathematics of exponential growth. The formula is n of t equals n0 times 2 raised to the power of t over g. Conceptually, this shows how a single cell doubles repeatedly over time. N of t is the total number of cloned cells. N0 is your starting number of selected cells. The variable t is the total time elapsed, and g is the generation time, which is how long it takes for one cell to divide. If time increases, the army grows exponentially. Let us put this formula to work with a practical scenario of a primary immune response. Suppose a single naive B cell is successfully selected by a newly invading viral antigen, meaning N0 is 1. We are given a generation time of 12 hours, and we want to find the total cell count after 120 hours, which is exactly 5 days. First, we find the number of generations by dividing the total time by the generation time, giving us 10 rounds of division. We then plug this into our formula, multiplying 1 by 2 raised to the 10th power. This yields a final answer of 1,024 cells. This physically demonstrates how a tiny recognition event scales into a measurable immune response. Building on our first calculation, let us now consider a secondary immune response, like when you are exposed to a virus you are vaccinated against. Here, you start with a larger pool of pre-existing memory cells, so N0 is 500. Memory cells react faster, with a generation time of just 8 hours. We want the cell count after 48 hours. Dividing 48 by 8 gives us 6 generations. We multiply our initial 500 cells by 2 raised to the 6th power, which is 64. This arithmetic yields a massive total of 32,000 fully functional immune cells in just 2 days. This perfectly illustrates the mathematical advantage of having a primed immune system. The staggering numbers we just calculated bring us to a crucial realization about public health. If the instructional theory were true, vaccines would not work because the body would not keep a memory of pre-selected cells. But because clonal selection is real, vaccines are incredibly effective. They safely introduce a harmless piece of a virus to select and multiply the right immune cells in advance. By starting with more cells and reacting faster, as we saw in the math, the immune system stops the disease before you even feel the symptoms. The clonal selection theory perfectly explains this life-saving phenomenon. We have covered a lot of ground today, so let us summarize the most important ideas. First, we saw the massive leap in human biology from the incorrect instructional theory to the proven selective theory. We learned that our bodies prefabricate millions of unique cells waiting for the right antigen. Once selected, that specific cell undergoes rapid, exponential growth to form a dedicated clone army, a process we can perfectly model with mathematics. This elegant mechanism of selection and expansion is the fundamental principle that keeps us alive in a world full of microscopic dangers. Great job following along with these foundational concepts.